Let us see about differential media. In differential media, we utilize this particular media composition when we cannot distinguish between the organisms which are very closely related to each other. At that time, we require certain parameters which can differentiate them properly, and we can identify them properly from the color of the colony or the reaction they have carried out in the medium state. So very first is we can find out difference between the organisms with this media with the help of visible color changes. Now these visible color changes will be because of the product they produce and their reaction with the indicator dyes which we have added in the media composition. So we will take examples of these differential media one by one and try to understand. Now these media usually contain more amount of sugars, different sugars are taken and their fermentation is observed, and on the basis of the fermentation they are able to produce certain acids, and these acids will be captured by the pH indicator dyes and they will give the color change, and on the basis of the color change we can identify the organism. Very first is McConkey's agar plate. In McConkey's agar plate, we have added lactose and neutral red is indicator dye. What happens that when organisms will grow, if they can utilize lactose and carry out its fermentation, they will give you pink color colony. The lactose non-fermented organisms will give colorless colony because neutral red will bind with a more amount of acid produced because of the lactose utilization by the organisms, and the colony will be completely pink. So here we can find out the lactose fermenter. In case of eosin methylene blue agar, we again have the lactose in the media composition, and we have two different dyes in the combination, that is eosin yellow and methylene blue. Now here we are going to differentiate in between Escherichia coli and Enterobacter aerogens, and that will be possible with the help of these two dyes. So if there will be production of acid in the more amount by lactose utilization, there will be you can say dark centered colony which will give you green matter exchange. So that will be Escherichia coli, and if the colony will only be having purple color and which are mucoid and bigger in the size, it is considered as Enterobacter aerogens. Now the next is MSA, that is mannitol salt agar plate. Now here we have added mannitol and we have added phenol that is indicator dye. So what will happen that mannitol will be utilized by the organism, especially gram positive one, that is staphylococci, and it will give you yellow color colony. So that will be the reaction of phenol red when it will bind with the acid produced at the end of the mannitol fermentation by staphylococci organisms. The next is xanthine, that is xylose lysine deoxycodate agar. Now these agar is specially made for identification of the members of family Enterobacteriaceae. Now here the xylose fermentation is given more emphasis, and xylose fermentation will be observed, and because of the different color changes and visual observation we can find out the members of the Enterobacteriaceae family. The next is TCBS, that is thiosulfate citrate bile salt sucrose. So this particular media contains sucrose as a carbohydrate, and it can give differentiation in between Vibrio cholerae and Vibrio parahemolyticus, and it will not allow the growth of coliforms and gram-positive organisms because of the composition ingredients. The next is blood agar. In case of blood agar, it is also differential on the basis of the zone of hemolysis we can get. Very first is alpha hemolysis, in which there will be partial lysis of red blood cells, and it will give you the differentiation in between organisms. In case of beta hemolysis, we can get clear zone, that is complete lysis of RBCs, and that will be done with the help of streptococci. In case of gamma hemolysis, there will be no change at all in the blood agar plate. So on the basis of these hemolysis zones and hemolysis observations, we can find out that which particular organism is grown in the blood agar plate. Okay, thank you.